Good morning, everyone, and thank you for attending my PhD graduate talk. My name is Sofia Hadir, and today I will present my PhD research entitled Root Growth and Below-Ground Interactions and Plasticity of Wheat Crops. To begin, I would like to place my research in the broader context of modern agriculture. In fact, modern agriculture faces several global challenges. On one hand, we need to produce enough food for a growing population. On the other hand, we are dealing with climate change. All this contribute to an increasing pressure on natural resources. Also, we have important challenges within agricultural system themselves. Crop production, for example, relies heavily on high fertilizer inputs. However, plants often use these nutrients or those fertilizer inefficiently, which leads to significant losses in terms of fertilizer. As shown in this figure, increasing fertilizer input does increase crop productivity up to a certain point, but nutrient use efficiency declines, especially when inputs become excessive. For that, we need to study more the resource processes that could be behind better use efficiency or an increased use efficiency of nutrients. The main mechanism that permits roots to adapt to environmental stresses is called root plasticity, which is the ability of a plant to modify its root system, morphology, and allocation in response to environmental conditions. However, most studies investigating root plasticity have been conducted under controlled or greenhouse conditions, which don't fully reflect the complexity of field conditions, for example, soil structure, and also the variability of weather conditions, but also the below ground interaction may have important effect on root system under field conditions. Especially in-depth field data about roots, growth, and plasticity remains scarce, and breeding also need the root data and the field condition. Also, crop model needs root data for calibration and validation. To address this, we designed a conceptual framework that permits us to explore different facets and drivers of root growth and plasticity and their field condition. The first is the biotic driver, which is the the intercropping, and the second is an abiotic driver, which is the nutrient variability. Both drivers influence root plasticity in terms of architecture and allocation and also morphology, and ultimately affect nutrient acquisition and crop productivity. This framework guided different studies included in my thesis. For this conceptual framework arise three main research questions. First, how do roots respond to intercropping, exploring the effects of neighbors, soil density, and genotype? Second and third, how cereal crops respond to nutrient omission, particularly nitrogen and phosphorus deficiency. And fourth, how sugar beet respond to nutrient limitation. Overall, the goal was to identify driver-specific and also crop-specific root plasticity strategies and their feed conditions. To respond to research question 1, we conduct a study on root growth and below-ground interactions in spring wheat faba bean intercrops. The main motivation is that intercropping, the practice of cultivating 2 or more crops simultaneously, has potential advantages for nutrients and land use efficiency. The below-ground mechanisms in such a root system are still understudied, especially under field conditions. For that, we conducted a campaign of root sampling in the spring wheat faba bean mixture trial in 2021. We focus on sampling root up to 1 meter depth using root coin method, and in order to discriminate between the species, we use the FTR spectroscopy. To study the impact of different soil densities and cultivar choice, we use 3 soil densities and 2 cultivars. The low soil density, the normal soil density, and the high soil density. And for the cultivars, we use the cultivar SO Ahab and the SO Annabelle. These combinations are encoded using these labels, which I will use in the following slides. One key result from this experiment is that intercropping increased yield efficiency, as indicated by land equivalent ratio higher than 1. This means that the intercrop system produces more yield per unit land area compared to sole cropping. These advantages suggest that complementarity between species may also occur below ground. When we examine the root distribution across soil layers, we observe differences between the two species. Faba bean tended to contribute more root biomass in the upper soil layer, as you can see here in the low total soil density, the normal soil density especially, and the spring wheat roots extended deeper. These patterns suggest that the two species may exploit different soil layers, reducing direct competition for resources, and this can be also can refer as to vertical niche differentiation. To study root mass advantages in intercrops, we conduct a comparison based on the expected from sowing densities and the attained root mass. We observed that the root mass in intercrops was higher than expected one in low and normal soil density, as you can see here. However, under very high total soil density, the attained values were lower than the expected one, which means that under low and normal soil density, we have a root mass advantage in intercrop compared to sole cropping, which can also increase the carbon sequestration. But under high soil density, we have root mass disadvantages. To study the root-root interaction more in details, we use the novel index, plant-plant interaction index, where the PPII is equal to PPII of wheat plus PPII of bean, and the PPII is the ratio between root mass of bean in intercrops and root mass of bean in sole crops, for example, for bean, and this is again divided by the ratio of plant densities in intercrops and in sole crops of the same species, for example, here bean. And just to understand this, PPII higher than 1 means facilitation, and PPII lower than 1 indicate competition. Our results show that soil density plays a key role in determining below ground interaction. As you can see here, in the very high soil density, we have a competition in both growth stages, but for the low and optimal total soil density, we have facilitation. So to sum up, the results indicate that there is a vertical differentiation between species, that total root mass is more sensitive to soil density than to wheat cultivar choice, and that interaction is density dependent. Very high soil density leads to competition, low to normal to facilitation. In addition to plant interaction, nutrient availability is another major driver of root plasticity. Different nutrients can trigger different root responses, and to investigate this, 2 studies were conducted on cereals within the long-term fertilizer experiment to Gobshof. The 2 studies were conducted on 2020 and 2022, and considering 2 types of cereals, the winter rye, the winter wheat, but also different depth of investigation. For example, we investigate only the topsoil in the winter rye, sorry, and we investigate up to 100 centimeter for winter wheat. We focus on nitrogen and phosphorus for winter rye, but for winter wheat, we used other types of nutrient omission, for example, the potassium, lime, and we used also a completely unfertilized treatment. And the sampling times, we had the higher temporal resolution of sampling in winter rye, while in winter wheat we only sample 1 to 2 times per growing period. So for the regarding the results of winter rye, as you can see here, the fully fertilized treatment led to the highest shoot biomass, especially around flowering. The nutrient omission treatment resulted in low shoot biomass compared to the fully fertilized treatments. In contrast, root biomass showed non-significant differences among treatments at the beginning of the growing season and around the early root stage, but the effects are more pronounced, especially for nitrogen omission in the late growth stage, as you can see here. The root to shoot ratio give insight into the allocation of biomass, and as you can see here, under nutrients, we have higher root to shoot ratio compared to well fertilized treatment. Beside investigating the root biomasses, also root morphological traits were investigated. We found that N limitation led to deeper angles here, which means the steeper angle, sorry, which means the deeper foraging, and that phosphorus limitation favors top soil foraging. The effect of growth stage on root responses to nutrient stress was also observed, and as you can see here, it's even much more pronounced than the effect of single nutrient omission. In winter wheat, nitrogen deficiency triggered the same strategy. Plant reduced shoot biomass but increased root allocation, resulting in a higher root to shoot ratio, for example, for nitrogen deficiency, and really very high root to shoot ratio in the unfertilized treatments. And this indicates an efficiency-oriented strategies where plant invest more in root land relative to shoot biomass. So what we can see here, nitrogen and unfertilized deficiency increased root to shoot ratio, but the depth distribution of roots remains comparatively stable, which means an intrinsic capacity to explore subsoil of winter wheat. So taken together, these results show that cereals mainly respond to nutrient stress through biomass allocation. For example, winter rye, we had nutrient-specific placement of root system, nitrogen, deep foraging, root to shoot allocation, and phosphorus, surface foraging through the increased nodal roots, and the allocation was also increased. We could observe this through the higher specific root length and the higher root to shoot ratios. For winter wheat, allocation compensation under nitrogen deficiency, because we observed a higher specific root length and also higher root to shoot ratio. And to sum up, nutrient omission means or leads to adaptive change in root morphology. Root plasticity was strongest under specific omission, but weakens under complete fertilization because the growth is collapsing. To complement this serial experiment, responses of sugar beet roots to nutrient omission was also investigated. The main motivation that the final system sugar beet is responsible for nutrient assimilation, but it's still under study. For this, we used data from a field experiment conducted at the experimental site Diekelshof, Germany. And the experiment included measurement of shoot and root rates across different growth stages using excavation and image-based root analysis. These slides summarize how sugar beet root traits responded to nutrient deficiency across 2 growth stages, the early growth stage and the late growth stage. Green arrows indicate an increase and red rows indicate decrease relative to the fully fertilized placement. Overall, nutrient limitation reduced shoot growth more consistently than root growth, as you can see here. And nitrogen deficiency showed the strongest response with clear reduction in shoot biomass and increased root to shoot ratio, suggesting greater allocation to root under stress. In contrast, potassium deficiency mainly altered root morphology, particularly through increased specific root length, which indicate a more exploratory root system. However, under complete absence of fertilization, both shoot and root performance declined strongly, especially at the low growth stage, as you can see here. Overall, this result shows that sugar beet exhibits adaptive root plasticity under moderate stress, but this response is insufficient under severe nutrient limitation. So, taken together, we could also find for the sugar beet that N omission increased the root to shoot ratio, indicating changes in biomass allocation between the below and above ground plants parts. Phosphorus decreases the root mass. Potassium is mainly decreasing the root diameter. No fertilization instrumental for shoot and root for both shoot and root growth. But we also found some experimental challenges related to the difficulties to sample the whole root system. In summary, we found that for the abiotic driver, especially intercropping, the low to normal soil density resulted in more roots biomass compared to the very high soil density, which means that the density affects root interactions in intercropping system. For abiotic driver, nutrient deficiency, so in cereals, phosphorus and potassium remains the major deficiency that affects root growth. Potassium and limonene are to a lesser extent affecting the root growth and plasticity of cereals. And we found also that cereals apply placement and efficient strategies to cope with stresses related to nutrient availability. In sugar beet, we found an increase in root allocation under nitrogen deficiency, but under unfertilization, we had a detrimental effect of this aspect on shoot and root growth than single nutrient omission. From those main findings, we could conclude some agronomic and ecosystem relevant implication. So the carbon sequestration via roots is increased under intercropping. We should also optimize soil density of intercropping system to take benefits of this system. And also the targeted, we should place nitrogen and phosphorus according to root foraging strategy. For example, nitrogen, it should be applied also in deeper layer, while in phosphorus we should focus on the topsoil layers. And for nutrient management in sugar beet, it is a key for optimal root growth in this cropping system. And we also found that plasticity partially compensates for nutrient stress but does not eliminate yield loss. Beyond the main results, this work also provides two additional contributions. First, it shows that root plasticity is strongly dependent of the developmental stage and the field conditions. This highlights that root's response must always be interpreted in a developmental context. Second, this work contributes to a field-based root rate data set that includes different types of crops, the sugar beet, faba bean, spring wheat, and also in intercrops, which enables model parameterization, for example, as in the work that's done by my colleague Derege. And finally, I would like to highlight key limitation and future research needs. Root plasticity research requires integration across multiple levels. First, quantifying root systems and their field conditions remains a major methodological challenge, which should be improved. Second, root responses are strongly influenced by interacting environmental factors, which requires studying interaction with water, availability, soil properties, and combined stress. Also, in this study, we only test one gradient of nutrient supply, either the fully fertilized or the omission treatment, but here it would make sense to test many gradients of nutrient supply to see the effect of nutrient availability on root growth. And also, future studies must include multi-year and multi-location trials. Also, we should use multiple genotypes to explore the genetic control of root plasticity. Thank you for your attention.